Thanks, Gil. So I officially started last June 1st. It's taken a while to get me settled in, and I have been trying to go around and get to meet individuals and other faculty in the department, in the Big Data Institute, and also in the Nuffield Department of Population Health. So actually, I thought it would be useful for me to spend some of my time in this lecture actually not talking about the latest, greatest things I'm working on, but also to talk to you about, where did I come from? People in neuroimaging, and I'm sorry for you guys, know what I have known for and what I've done, but actually a lot of people don't know, and it's very possible that some of the things I've worked on are actually very useful and relevant to some of the work that people do. So I'm going to take a sort of slightly informal view of this and say, okay, how did I get here? Well, so I started by doing a computer science degree, and then I had so many nights in the computer cluster with no sleep, I just said, I do not want to spend the rest of my life in front of a computer. Little did I know. But anyway, so I changed from computer science to statistics. It was all mathematics, and it was a small move. And I finished that, and that was good. But actually, my true passion was photography. I'd been the photography editor for the newspaper, and that's what I wanted to do. And to great distress of my parents, I actually abandoned science and worked as a photographer. I dug these up last night. I was working as a professional photographer in Pittsburgh. That's a steel mill. That's a juvenile detention center in West Virginia. I don't know, what was that? These are all the test shots. So as we were setting up the lights, did some pictures from some bands. But then the student loans came due at the end of the summer. And fortune would have it that Mike Escobar, a professor at Carnegie Mellon, called me and said, there's a job at the hospital. It involves images. It's like, that's great. I know about JPEG, PIC, TIFF. And he's like, well, no, it's more like MRI and PET, but are you interested? It's like, and very reluctantly, I said, okay, I'll take this job. And so it was a job as a programmer at the University of Pittsburgh Medical Center's PET facility. And I didn't know anything about medical imaging, but it turned out that there was sort of a great need for statisticians there, and so even though I was a programmer, I was immediately called on to sort of do statistical thinking, and also due to my great fortune, the newly recruited faculty members had tight ties to London. And in London, there was this faculty member, Carl Friston, who had just developed statistical parametric mapping, which was being sent around by photocopies of photocopies and on tape discs, tapes like that, if anyone remembers those. I think it's a 1/4-inch linear tape. And so I got there, and my job was figure out this SPM thing. And then luckily, they then sent me to London. And it was in London that I got to meet Carl Friston, but he said, why don't you work with this graduate student, Andrew Holmes, who was a student of Ian Ford's and Carl Friston's, and so I went to the cyclotron unit, visited them, and we really hit it off. And so we decided we were going to implement, instead of SPM, oh gosh, font problems. Yeah, that's statistical parametric mapping. That was some very nice ASCII art of the letters SPM, but that's lost there. And so we developed SNPM. There's Andrew. This you can imply any kind of attitudes about Americans. This is actually at a Prince and Paupers party. We were dressed as paupers. But he visited me in Pittsburgh. I visited London repeatedly, and we developed this toolbox that was first released in 1997, and we've continued to develop. He's actually left academia, but I've continued to develop this. And sort of started a long-term interest in resampling methods for brain imaging inference. So then I realized I had a lot more to learn, so I went back to Carnegie Mellon for a PhD in statistics, still working on PET data. Again, through connections at the PET facility, someone said, you should go visit University of Southern California. There's a really smart guy there, Richard Leakey, and so I did. And there I went to California and I made movies, as you do. So this, usually with PET data, you just get one static picture of the brain or a series of sort of like histograms. And so in my thesis work, I actually developed methods for a spline-based spatial temporal reconstruction of the brain. And so this is actually watching a O15 bolus of radioactivity come up through the carotid arteries and perfuse the brain. So that was what I got out of Pittsburgh, and what I learned from this is that having seen the impact of, just in that time, of Carl Friston's work of giving away this SPM software, but also answering questions. I would see investigators actually call him on the phone randomly, cold, and he would actually answer their calls and help them. And it really actually made a difference because more and more people used it, and he made open software that anyone could use. And that was something we were able to bootstrap off of when we made our SNPM toolbox. And also the great thing of actually traveling the world and actually getting paid to do it, and doing science at the same time. So I really enjoyed that. So then I took my first faculty position at the University of Michigan in the Department of Statistics, and there I focused on MRI more, and inference on MRI. And so this was the time that there was, we were first really coming to grapple with multiple testing in a big way, both due to brain imaging and things like microarray and gene expression. We have this problem that if you have 100,000 tests and you use a conventional 5% threshold, you're going to get 5,000 false positives on average. And so to deal with this, you need to come up with the right measure of false positives in this multiple testing setting. And so the two ones we generally end up using is the family-wise error rate, but at the time there was a newly proposed method called the false discovery rate, and that's the proportion of false positives among all the positives. So we put these to use and implemented them in SPM, and here you can see that this is just an example of an fMRI experiment. Here, if you don't do any thresholding, you just see, well, there you get a value, you get one test statistic for every point in the brain, and of course you don't know what's real and what's not real. So we do some sort of thresholding, and if you use the standard methods at the time, random field theory, you get this result. We were able to improve on that with permutation, and then if you used a false discovery rate, you got yet more sensitivity. Of course, it's a bit of apples and oranges. You are accepting some false positives in this map here, but they're in some measured control. And this is something that actually I came back to in my research again and again, because this is kind of surprising. If you know anything about non-parametric methods, usually when all the assumptions are satisfied, there's no extra win. I mean, a non-parametric method can be no more powerful than a parametric method when all the assumptions are satisfied. So this is kind of funny that, especially in these small data settings, there's actually only 12 subjects, we're actually doing better with a non-parametric method. And that's something that I've sort of revisited and tried to figure out what is the origin of that difference. And we eventually pinned it down to some violations of some assumptions that people didn't realize were hidden in the standard random field theory methods. And this was our first foray into that work, showing that these methods that everyone was using, this is the random field theory method, actually were incredibly conservative. This is under Monte Carlo simulations. Even when all the assumptions are satisfied, they are conservative. And the permutation methods have no problem with that. And this is either with a voxel-wise approach or a cluster-based approach. This is where we assess the significance based on the extent of the spatial extent of the signal. So this is sort of my first work with inference and focusing on sort of what are the valid but yet powerful methods that we can depend on. And then another thing I'll come to, I want to mention is something that was actually sort of not that deep statistically, but turned out to be rather important. So one of the things that psychologists often need to do is try to figure out where is there consistency among a set of inferences. So one example is you might be doing a working memory experiment, and you might be shown a list of letters, and this is a classic NVASC. So for example, you might be asked to look to see when is there a series of letters that are separated by one interval. And I think actually there aren't any here. But the point is you're supposed to remember with the previous letter, or maybe it's the previous number, or it's a previous shape. Now, is the substrate in the brain that's responsible for shape memory, number memory, and letter memory, are those all the same, or are they different? So a question people want to know is, where in the brain am I activating for all three of these? And so this is, I don't want a meta-analysis of any. I want a response. I want to know where do all three of these respond. So the idea was proposed that we should be using the minimum statistic. So if I have three things, I'm going to take the smallest of them. And certainly if the smallest of three test statistics is significant, then certainly all of them should be significant. So Carl Friston and Keith Worsley proposed a method based on this minimum idea. And so they actually have some very elegant mathematics that studies the properties of a minimum of a set of statistic images, a minimum random field. And then they conducted inferences on this by comparing under the null hypothesis, where there's no effects present, versus the alternative. And somehow they never sort of caught this thing that actually this is not the right null hypothesis, actually, because if you're trying to do a conjunction, you're trying to say that all the effects. So actually what you wanted to do is to say, no, I need a union of all the null hypotheses. So yes, not all effects present, and I want to reject that in favor of all effects present. And so we showed rigorously that actually, and it's kind of embarrassing, the right thing to do here is you just actually make sure that all tests are significant at the usual level. This also I've later learned can be known as a union intersection test. But this is what taught me is that this was something that was not very sophisticated, but it was brought to me by psychologists who said this just doesn't make sense. There's something missing in the connection here. And it's when I realized that being integrated and closely working with the psychologists that are using the tools that I was interested in is very important and, well, fruitful. So this paper, which again is not very sophisticated mathematically, is become one of my highest cited papers because we corrected this approach that was faulty. So what I learned in Michigan, I'd say, is the rewards from working closely with people that are actually in the trenches using these imaging methods. I at that time continued to develop the SNPM toolbox and other tools, and supporting them. And again, that it doesn't have to be the most highfalutin statistics to be impactful. So then what happened is that I met my wife. This is my wife. That's the mid-sagittal plane of my wife when she was young. And so she is actually from Germany, and so when their job opportunity came up for an English-speaking European country, I actually said, oh, that sounds interesting. That's what she looks like on the outside, if you're interested. So that job was at GlaxoSmithKline. When people meet me at parties, oh, you work at the Great Western Way on the way to the airport in Heathrow. I was like, no. I worked next to the Wormwood Scrubs prison, where the GlaxoSmithKline Clinical Imaging Center was located. And it was quite a bold move. Drug companies generally do not touch humans themselves, right? They always use contract research organizations. And GSK, in an attempt to accelerate the development of drugs for psychiatry and neurology, said, no, we're going to bring all this in-house. We're going to bring in the cyclotrons, we're going to bring in the PET scanners, we're going to bring the MRI scanners, we're going to do it. And it was a collaboration with the MRC and Imperial, but it was wholly, the actual floors that were ours were wholly ours, and we were wholly responsible for them. So I was brought there to work on imaging genetics. So basically the intersection of brain imaging and genetics. I was given the title of Director of Modeling and Genetics. But literally on the first day on the job, Paul Matthews, my boss, said, don't worry, but we've just fired 500 geneticists, and genetics is kind of a bad word in the company right now. This was when this was Alan Rose's group in South Carolina. So this was a lesson about the volatility of corporate world. But to Paul Matthews' credit, he actually did protect me, and I did continue to work on brain imaging genetics. And one of the first things I did was looking at this glycogen synthase kinase pathway, which I cannot believe spells GSK. This was my first imaging genetics paper. It has nothing to do with GlaxoSmithKline, but I had a postdoc who was a geneticist, and she was really interested in the Wnt signaling pathway, and she thought the best way to access this was through GSK3-beta. So this was a subset of a traditional case control study. So out of the 1,000 cases, 1,000 controls, we had 200 with brain imaging on the cases, 200 with brain imaging on the controls, and we used something called voxel-based morphometry to measure the gray matter. So this is a way of studying anatomical differences all in the same space. You basically warp the subjects to be as similar as possible, and then you put back in the anatomical differences in the atlas space. So we were searching for brain volume associations. And when we did this, we found both main effects and interactions with this gene. So we had to search over 15 SNPs, and so we did have to protect ourselves for that multiple testing problem. And we also had to account for something called non-stationarity. It turns out the smoothness in VBM data is spatially varying, much like LD varies in the genome, but it's likewise in brain imaging data, the spatial dependence varies. And so we had to account for that. But we did actually get a significant hit correcting for all these, and also a significant interactions demonstrating that there was something changing in the hippocampus, and that was explained by this variant, this genetic variant. And this, of course, is not crazy that people had previously seen that there are structural differences in the hippocampus with people with populations with depression. So this is just an example of sort of one of the first imaging genetics associations we've done. Now that is what I would call a candidate gene study, and there was a movement away from them because there's always, well, okay, after we did GSK3 beta, what would be the next gene, and what would be the next gene, what's the next gene, and then how are you actually getting confidence that those results are real after you've sort of tested every gene in the genome. So one of the things I developed there was with Giovanni Montana at Imperial was to try to grapple with the problem of, can I try to associate all genetic markers with all brain imaging voxels? And if I did that, I would have to deal with a huge number of voxels by number of genes correlations, and there's just really, we don't think we could actually realistically estimate them. But importantly, we don't think they would all be non-zero. So the approach that we took was something called a sparse reduced rank regression, where we said, okay, what we're looking for is some small linear combination of a handful of SNPs and a handful of imaging measures. And that's the way we're going to constrain this to make this practical. And this reduces the dimensionality and the complexity that gives us a hope of finding this. So we showed that this approach was more sensitive than the naive, just everything with everything. And then he went on with the same student to show that we can use this. Now, there are some more tricks because we're no longer in the traditional hypothesis testing framework of classical statistics, so we needed to mix in some machine learning techniques. So basically doing selecting the most important parts of the selecting the most important parts of the image to feed into the association, and then choosing what is the degree of sparsity that is the right amount. That is a free tuning parameter that you have to set. And they use stability selection and found both some genes that are well known to be associated with gray matter, with Alzheimer's disease, but also some other ones that had not been previously reported. And then also one thing that I did as I was leaving and transitioning was adaptive design for clinical trials. This is a question that Paul Matthews had put to me of basically, well, we know that we pay a penalty for searching over the brain for activations, so could I collect a small number of subjects in a drug trial to figure out which are the most important regions and then carry on with a second phase where I just look at those important regions? And this is something that's fundamental because in general, in a drug trial, you can't look at 100,000 voxels. You have to commit. But even in brain imaging, when I went to my colleagues at GSK, it's okay, we're not doing voxel-wise. Tell me what is the ROI you're interested in? And they invariably come back with 20 ROIs or 10 or saying, no, we're not sure which. So that was something that we did to try to say, could we do sort of a pilot and basically try to adaptively find some weight, some linear combination of all these regions that's optimal. And there wasn't an entirely simple story that came out of this, but just to say that if you're predicting the power, you have to predict the fact that every time you run this adaptive design, you're going to get a different outcome. You're going to get a different estimated number of n, and so all my power predictions here have confidence intervals on them. But I think this is the most interesting thing. If you fix a total sample size, then you can find what is the sort of the optimum. So if I only have 14 subjects, which unfortunately in early stage clinical trials is not totally crazy, you can say, well, but you should probably take about a quarter of those and use those quarter of those subjects to develop a pilot study to estimate what are the important regions, and then use the rest to determine the outcome. So GSK, I definitely was impressed by the quality of people. I think I had this impression coming into industry that it would be kind of the science would be softer, and if anything, it was much harder. They had finite budgets, big budgets, but finite budgets, and a nearly limitless set of candidate sort of marker genes and pathways that they can investigate and candidate drugs. And they were always trying to use the best sort of evidence possible to decide which of those drugs should they advance to the next stage. So I was impressed by the people there and the methods they used. However, every 6 months there's a change in priorities. Something is going to be different. You shouldn't be working on, now we're all going to work on this, not that. And so I really wanted to get back to academia. Also learned that just clinical trials are hard to do. So I was recruited by the University of Warwick to join the Warwick Manufacturing Group because at the time the Warwick Manufacturing Group thought they wanted to get into neuromarketing. This is the use of fMRI to decide what's the best way to develop an ad campaign or make decisions about product framing. Fortunately, that effort died after I was recruited. So I was recruited, and I insisted on being joint between WMG and Department of Statistics. That happened right soon after the economic sort of crunch, 2008, and so that didn't go forward. But I ended up there, and then I wrote a fellowship to the Wellcome Trust on meta-analysis and data sharing. So I'll tell you a little bit about that meta-analysis work. In brain imaging, the form of meta-analyses are fundamentally different than what you've seen in other settings. Usually meta-analysis is I have a sample mean from this paper, a sample mean from that paper, and a sample mean from that paper, and how do I bring them together? In brain imaging, we have this unfortunate situation that the image data of the results has not been shared. So for the past 30 years, all that you can depend on is a table of XYZ coordinates of where was there a peak, a local maxima in the brain in the statistic image. So all we have for each study is, oh yeah, I would love to have this map of 100,000 voxels. It's gone. Now, things are changing now, I can happily say, but in the past, all we have are these points in space. And so the challenge in a neuroimaging meta-analysis, or what we call a coordinate-based meta-analysis, is how do we synthesize these collection of point patterns to see where are the locations of typical patterns. So again, we would like to do the image-based, the intensity-based meta-analysis, but we just don't have that data. So one of the things I worked on actually has this really fun motivation. Actually, it's kind of sad now. This is back when Hillary Clinton and Barack Obama were in the primaries and battling it out. And there was this scientific result in the New York Times about someone who took 20 subjects, put them in a scanner, and then published these results on the op-ed pages. And they said that voters who a priori disliked Hillary Clinton exhibited significant activity in the anterior cingulate, an emotional center, activated when one feels compelled to act in two different ways but must choose. So you can see what they've done here. They've taken an image of brain activity and they've made a conclusion about what the person is thinking. They're emotionally conflicted when they see Hillary Clinton, right? But this is faulty logic. They're basically saying, in the past, we have seen that when we have conducted carefully designed emotional conflict resolution tasks, we have seen anterior cingulate activation. We have now seen, when presented with Hillary Clinton, we get anterior cingulate activation, ergo, Hillary Clinton induces emotional conflict. And of course, it doesn't work. You cannot reason to the consequent. This is faulty inference, and so we can't sort of turn around the conditional probabilities. So what do you need to do? You need to be able to consider all of the different possible outcomes. What are all the different types of experiments that could have given rise to an anterior cingulate activation? Well, no one person can do all those experiments, but you can do a meta-analysis. So I was involved in work with Holly Arcani, who was at the time at the University of Colorado. He's now at University of Texas. Who said, okay, we're limited by how many people, how many grad students can code up these papers. So what we're going to do is build a lexicographical analysis that will just analyze the HTML of papers and note what are the words you find there, and then also automatically harvest the XYZ coordinates. So this part is just basically automating what people have been doing manually, but also sort of just coming up with decisions about what is the topic of the paper based on word frequencies. And so the traditional sort of setting that we do a meta-analysis is we collect all the meta-analysis, the papers we could find doing pain, and we say what are the typical patterns of all the studies that have studied pain. But in a reverse inference, you can say, okay, here's my one brain activity pattern. What was the most likely experiment to give rise to this brain activation pattern? And so this is what the brain imaging people have called reverse inference. And there's been sort of traditions of people sort of informally doing it, as in that New York Times article. And instead, if we have all this information collated, we can then do this and make a formal assertion of probabilities, saying if this is what we see as an input, yes, there's some probability that it's one of these other classes, but it's most likely to be, say, pain here.
Where would you get your priors for that?
So the priors, well, your priors, you'd say we generally use flat priors, and then we just try to have it driven by these data here. Or do you mean by priors for the different
Yes, in your reverse.
Yes, so that is an issue. So you have to. We certainly can say you give me a word, and I'll give you all the papers that have that word in more than 1 in 1,000 occurrences. But yes, you're right. It is up to someone to come up with the list of those topics. And so far we just sort of used equal prevalence, but that is a serious problem because the prevalence is going to be affected by what are the hot topics, right, for people to study. So, yeah. So, and then the sort of the footnote to that Barack Obama Hillary Clinton example is that actually from this paper that we did on neuroscience, if we just said actually just overall where are their results reported in the brain. So this is just really basically, if you want a histogram, over all studies that we saw, over the thousands of studies that we harvested from literature, where do we get points? Well, actually, the anterior cingulate is one of the most frequently activated points in the brain. So basically, if you only tell me, oh, I saw the anterior cingulate activated, I probably can't tell you very much. I could probably point to about a fifth of the literature that may have given rise to that, but there's actually, it's a highly non-specific area. So this work was done with rather simple, naive Bayes approach. Where we just divided up the brain into voxels. The statistical work I've done on that has been trying to use more sophisticated spatial point process methods. And I know there's people here who do the disease mapping that use similar log Gaussian Cox processes. Very similar techniques are used. Here we're using a Cox cluster process to try to account for the clustering that we saw in this particular pain emotion meta-analysis data where there's lots of clustering around these amygdala areas. We used a hierarchical model where we could distinguish between how spread out are the foci in a typical paper versus where is our inference on about where those foci cluster. So you can kind of think of this as kind of the standard population standard deviation, where this is the kind of population, the spread is a population standard error. The more subjects, more studies, will get tighter and tighter inference, whereas with more studies, hopefully this will stay about the same. And then we tried our hand at a small scale reverse inference, just saying, could we distinguish what are the 5 different types of emotional experiments that went into this meta-analysis, and found that we could do pretty well with our spatial point process model, getting an accuracy of 80% when chance was 20%. And the naive Bayes approach that we used previously is above chance, surely, but only much worse classification rate. So now, finally turning to big data. So is fMRI big data? Traditionally not. This is from a review paper we did last year, where we did another meta-analysis just harvesting the sample size by year. And so these are the median sample sizes by year. We changed methodology here, but you can see that they seem pretty consistent. So only in 2010 did we sort of pass the n equals 20 barrier for the median sample size. So this alone is a motivation for why meta-analysis is so important when each individual study is so small. And showing that really, I mean, some of these studies were, would have required enormous effect sizes. And if you know, in a typical social science study, you have an effect size of, you'd be very, very happy if you had an effect size of, say, 0.3 or 0.5. Now, it's not totally crazy. This, you might say, how does this, it must be fMRI is all bunk. No, but do consider that fMRI is a designed experiment. This is not a natural experiment. You're just surveying people on the street. You are putting people on a scanner and you are poking them. I mean, sometimes burning them, an pain experiment, but you are inducing experimental variation. So it's not, so it is a steep ask, but these are not crazy. We do attain these, and we did a review of data and found that we do actually get samples, effect sizes that large. So the first, I would say, bigger neuroimaging data project I was involved in is the Human Connectome project. This is based in America at the Washington University. The FIMRIB group was tightly involved in this, collecting state-of-the-art data to understand how different parts of the brain connect to other parts. This is a population sample, a proper population sample where people of a given birth cohort were invited from the Department of Records in Missouri. So this is should be a quite representative sample, and we're selecting from that cohort those with twins and siblings. So the target was trying to find about 300 families of size 4. And so we have cutting-edge imaging and a lot of behavioral. I thought this was a lot of behavioral variables until I saw the UK Biobank, but it is still a lot. And all this data is openly shared, and it's really been incredible for sort of developing the methodology of this of brain connectivity. So I'll just skip that. So what I was first charged with was thinking about heritability, because of course we have twin data, so we have twins and families. And one of the things you run into is that the estimating heritability can be sort of a tricky thing. The likelihood can be very ill-conditioned, and even though you say 1,200 subjects, well, as the data was being collected, we didn't have that many families, and we started sort of with small data, and we wanted to get something reasonable. Also, we wanted something that would be reliable if we were trying to do it on connectomes. Now, in the worst-case scenario, you say 100,000 spatial elements. The dense connectome is the 100,000 by 100,000 correlation matrix. And you might want to say, you might ask, well, how heritable are the edges in that matrix? So we wanted something fast. And this is also, I wanted to illustrate some I think, a great way that Steve Smith and I have worked together. This is a traditional way. If you've ever done a twins analysis, you take a phenotype and you look at the correlation between the monozygotic twin pairs and the dizygotic twin pairs, and you basically use the difference of these correlations to help let you learn about heritability. Steve Smith was working with all the imaging data, and of course, so for each subject, we have thousands or hundreds of thousands or millions of measurements, and he said, well, why don't we just correlate twin pairs individually? I mean, it's not something you'd ever do in a traditional quantitative genetics analysis. Typically, you just have one measure, so you couldn't do this. But his sort of just intuition was, why don't we just do this? Why don't we take twin pair 1, twin pair 2, and just note whether they're MZ, DZ, and then for each twin pair, we get a number, which is the correlation of their high-dimensional phenotype, and then basically make a box plot. And surely, if we see a difference in those correlations, the correlations of high-dimensional phenotypes between twin pairs, it must say something about heritability. And then that's where I came in with working with my graduate student, Shui Chen, saying, well, what does that mean? I mean, because it's not a standard measure at all. Does this mean something? And so we were able to work out, skipping all the details, that actually, yes, if you can work out what that these things are, not heritability per se, but a weighted average of heritability plus a contamination term, if you want. This is something called an endophenotype ranking value. It's a weighted weight of heritability and a genetic correlation. So actually, yes, this thing that Steve has been computing, the difference of these means of these two different groups, does have a concrete meaning. It is the variance-weighted heritability shifted and corrupted, basically, by some measure. However, one thing you can show is, well, what if you are just sort of looking at the significance of this? Well, this ERV thing here, well, this depends on this genetic correlation. The genetic correlation is going to be 0 if the heritability is 0, and if I'm sorry, this here, the ERV will be 0 if the heritabilities are 0. And likewise, this phenotypic adjustment sort of attenuation term, that too will be 0, because if you have 0 heritability, the genetic correlation should be 0. So it turns out that even though this is kind of a crazy, sloppy thing, it's really fast to compute, and it is a valid test for 0 heritability over all the elements of the high-dimensional phenotype. And so we put that to work in saying, well, maybe if we're just interested in ranking the heritability of various phenotypes that are high-dimensional, would this be useful? And it is. So we did it the fast way, we're calling it aggregate heritability, and the slow way, which was using a best practice maximum likelihood estimate of the heritability on each element and then averaging those. And you get decent agreement, correlation of 0.81, but importantly, the ranking is quite similar. So if you pick the top 4, that's the top 4 in either approach here. And if you compare this to the variance-weighted heritability, the correlation is even similar, which makes sense because that's what we worked out the aggregate heritability is. And so that's one example of something we've done to sort of speed things up, computational speed-ups to sort of answer questions that may not be practical using sort of the standard genetic methods. The other thing I want to talk about is something I'm very interested in, is basically how big data is forcing us to think differently about the standard tools that we use. So brain imaging, in fact, my entire career has been based on null hypothesis testing. What's the threshold, Tom? What's the right threshold? Is it valid? Is it conservative? Is it liberal? However, there's already been a hint that this is maybe not so interesting going forward. So one study was done at NIH where they took a subject, I don't know how they got them to do it, but they put them in the scanner 100 times. And then they looked to see where in the brain is there significant activation with a fairly simple task. And the answer is everywhere. They were able to detect systematic variation related to the experimental design at basically every point in gray matter. So this is illustrating what's sometimes referred to as a null hypothesis testing fallacy. With enough data, you will eventually reject the null, because the null hypothesis is in a way a crazy statement, right? It's saying that the effect is 0.00000, I mean, absent, totally absent. So that got me thinking that, of course, if we're going to have 100,000 subjects, we're going to be running into the same problem. So what can we do? And so this is some work I've been developing with my student Alex and a collaborator at University of California, San Diego, Armin Schwartzman, to basically develop spatial confidence sets that work even if there is signal. And the idea is you pick your meaningful threshold. That might be, say, a 1% bold change or an effect size threshold. And then we work out what is the appropriate threshold up and down such that we get three, basically, sets of thresholds. We get a inner threshold where we're confident that the effect is bigger than this. And then what's also really nice, and you never get in the null hypothesis testing framework, anything outside of the blue we can be confident is smaller than this. So we basically, in a way, get to prove the null or say that the null hypothesis is true, but of course it's not the null. It's just saying it's a statement about the true effect magnitude relative to this meaningful threshold. So we applied this first in a relatively small example of the human Connectome project on 80 subjects with a 0.5% bold change effect, and then a lower one. So if we are saying that our scientifically meaningful percent bold change is 0.5%, we can see, okay, this is the amygdala. I have confidence that this. These are faces. These are sort of fearful and angry faces versus shapes. So this is both activates the faces areas of the brain and also areas involved in emotional processing, which is the amygdala. But we can see that we can be very sure that we're getting at least 0.5% change in the red areas. But if we wanted to be sure that we're getting 0.5%, then we have these larger regions here. And then just to show that if you had a lower threshold, you can say, well, I'm really not. If I want to be really confident that I have a 0.2% change, I can't be. I really can't rule out all these brain regions. They could have a 0.2% change, not sure. But if I'm trying to say, is it a 0.5% change? Yes, I can be sure that only in these areas are getting the 0.5% change. And then also applied this to the UK Biobank recently. And so here we just took the 1,000 subjects and 5,000 subjects. So these are all the brain regions that are significant at Bonferroni 5%. And we could do better if Bonferroni will be conservative, so there's actually probably surely more regions where we would reject the null hypothesis. But now if we're actually saying, no, I want to know where is there at least a 0.2% change, and now I can get these bounds here. And now you can see with 1,000 subjects, we actually have some high spatial precision. So this is actually good because it actually is telling us that we are probably reaching a point where you just threshold. You pick your threshold, and that's going to be it. Because eventually, so here the boundary had 12,000 voxels in it. So the boundary between the blue and the red is 12,000, and with more and more subjects, that boundary is coming down and collapsing. Now, this is not to say there's no point in this. Of course, if I asked a yet more challenging threshold, if I said, actually, I want to be sure where am I confident that there is a 0.1% or a 0.01%, well, then, of course, these boundaries will be wider. So this is the sort of thing I'm sort of excited by. These are places where the traditional tools just break down and aren't meaningful, and we need to build up a new set of tools that are going to be useful for this sort of data. Then just drawing on some insights that from a recent summary paper that Steve Smith and I wrote on challenges in big data neuroimaging. One of them that kind of surprised me is that if you actually just ask the question, what is the power I have to detect an association when I have many, many tests, in this case, many, many features, because once we acknowledge that we have all the variables in the biobank, but we also might be interested in testing for associations between them, and so the number of tests that we might want to do is just growing and growing. But if you say, okay, I want to be powered for a 0.1%, say right here, well, if you just have one test, you need a lot of subjects, 7,000. But what's fascinating is that actually, if you say, well, actually, yeah, I want to detect a 0.1% change, but I want to look at 10,000 tests, okay, so you need 30,000. Again, these to me would be sobering numbers, but again, we know these are within reach with biobanks. And what's really remarkable to me is that if you look at this, it turns out that a squaring of the number of tests only results in a doubling in the n needed. So this is something that surprised me, that actually that there are two things that are vying for each other. You have the penalty for searching over more and more tests, and you have the gain from having more and more sample size. And it turns out n wins, basically. So that was surprising. We didn't expect that. And then the other thing, we just wanted to get ourselves depressed, and so we just itemized all the different ways things that can go wrong. And so this is something that, my methods are focused on providing inferences that you can trust based on the data and based on assumptions about the data. But what they're not telling you about are confounds. So you could find an effect that I say, yes, that is a reliable statistical effect. But it also could be due to head motion, which you might mistake for an effect of attention deficit hyperactive disorder, because kids in the scanner tend to move more and will have more motion artifacts. We are seeing that changes in the way you breathe. If you breathe very systematically and steady as opposed to, that can actually cause artifacts and result in systematic differences in the image data between individuals depending on these breathing patterns. Age, and then the really sort of sobering one is scanner hardware. We're kind of aware of that, but on long-term, large-scale projects like the UK Biobank, we have to worry about things like scanner operator consistency. And these things actually they've detected effects over time of, if it's not an individual scanner, operators, just the way in which they conduct the scans have changed slightly, and that results in systematic changes in the imaging. So so that's everything I wanted to say. I wanted to give you an introduction of who I am, how I got here, and then say a little bit about neuroimaging data. They're basically kind of three sort of interesting things, I think, are the computational aspects. How can we take things that we're already doing and make them more efficient, scale them up in ways to make them practical for the large-scale data? Then there are kind of the fascinating sort of statistical things, problems that weren't there before, things that could not have been contemplated when you only had 2- and 3-digit sample sizes. The new statistical methodology that just wasn't an issue before. And then finally, sort of, and this is where I'm sort of really looking to learn from my colleagues here, is basically how we grapple with these epidemiological concerns and figuring out whether we're seeing a real effect just like that is a confound. So I'll stop there. Thanks for your attention.